Hi everyone, my name is Tom Gawriluk, and I'm the Spinal Cord Program Lead for Novoron Bioscience. Thank you to the organizers for giving our early stage company this opportunity. And I'm going to talk today just about who we are and how we are developing a spinal cord injury treatment for patients with spinal cord injury. This is our team. I want to start off here. We're actually downtown San Diego. This is our window outside the lab. And so Padres fans going to a game, they can actually see the logo and give us a call, look us up. But importantly, I just want to point out that we have a funding partner, Two Bear Capital. They've been very active in terms of us developing our spinal cord injury program, and they're open and happy to having introductions to kind of broaden their own impact. Other people I want to introduce here is the co-founders, Travis Stiles and Sean Garr. They're here in the audience, and so if there's any questions or you want to follow up, please find us after the presentation. And so we are a seed stage biotech that was founded around a technology out of UCSD to develop a spinal cord injury treatment. Pre-2020, we were really focused on grant funding, and that kept us small, but we actually were able to accomplish quite a bit. In 2021, we partnered with Two Bear, and that allowed us to expand our team and really kind of focus on how we were managing our projects and scale those projects. Just shown here is kind of a network of our projects, with green being Novoron, gray being our academic partners. And so you can see Novoron San Diego, this is our kind of hub for innovation. Then we have two contingents, one in Australia and one in Lexington, Kentucky. And they're working alongside our academic collaborators. And so as an example, so I'm in Lexington, Kentucky with another scientist, Michael Cabrera, and we are performing our preclinical animal models alongside Warren and John's groups at the University of Kentucky. And so this gives us a unique opportunity to really understand those models and also kind of drive the research a little bit more, and also keep that institutional knowledge within Novoron. And so as we find issues with those models, or if we find issues with our drug delivery paradigms, we can directly address those with the idea of being able to push that into a clinical setting. And so Novoron's focus is low-density lipoprotein receptors and being able to create a drug against them. LDLRs are a vast family members, and there's a lot of therapeutic potential in these family members. The main challenge has been being able to target a single drug indication, and we're focusing on LRP1 because that's really how we got our start, and it's an important molecule for spinal cord injury and other things that we're potentially working on. Our solution is really twofold. So one, if we antagonize LRP1, there's always the risk of antagonizing all these other family members because those domains are rather similar. But we have a comprehensive antagonist that binds to multiple domains at once, really shutting down LRP1. And two, we have proprietary platform and tools to really develop a small molecule program to target specific ligand drug domain interactions. And so for the context of spinal cord injury, I'm going to focus on our comprehensive antagonist, specifically delivered to that spinal cord injury. And so the problem becomes, immediately after spinal cord injury, we have axons that get sheared, we have cells that die, and we have myelin debris that is present. And so myelin debris is just these vesicles that have ligands, and those ligands are now going to start binding to receptors that normally wouldn't happen in healthy tissue. And myelin, we know, is an inhibitory factor for not only neuron growth, but neuron degeneration and the ability for axons to regenerate. And so what we're doing is we're delivering our LRP1 antagonist directly to the spinal cord after spinal cord injury, and we're going to antagonize LRP1 to basically allow those neurons and other cells to ignore the myelin and restore the natural regenerative capacity of the spinal cord. We've been able to show this multiple different ways. This is just one example, some cells, some neurons in a dish, but you can see here that on the right-hand side, with the, it doesn't show up, but yeah. And so cells grown in the presence of myelin, they have inhibited axons. And if we grow them in the with the presence of a off-the-shelf comprehensive antagonist called RAP, we've actually restored that axon growth. And so we've been able to show this in multiple cell types from multiple species using both genetic approaches and antagonist approaches. And so this has validated that LRP1 is an important target in this system. And and so that's really how we're moving forward with this. And so how does this work? This slide incorporates a lot of our own work and others' work. And if you want to get into any of the nuances of this mechanism, please find me afterwards. But basically at the top here, we have some myelin debris and we have some inhibitory myelin components. They're going to bind to their respective receptors on either a neuron or an OPC. And there's going to be a complex that gets recruited, and that's going to contain P75, LINGO, and LRP1. At that point, this entire thing gets endocytosed into the cell and becomes a signalosome. And I want to point out that this is a convergent point up here where LRP1 is binding to these other things. They're very disparate pathways. I'm just showing 3 here, but recognize that all of these things end up being in this signalosome. The signalosome then becomes a cell signaling center, and downstream of that is activation of RhoA, which may sound familiar to some people in the audience, but we know that RhoA is a hallmark of neuron or growth cone collapse and neuron degeneration, just in cells in general. With the addition of NOVO118, which is a comprehensive antagonist that we have developed, we're actually going to bind LRP1, and now this complex can no longer be endocytosed into the cell. And that's going to cause basically increased axon regeneration potential because we're not activating RhoA. This is just some data here on the bottom, really just showing that in the presence of 3 different myelin inhibitory factors and our NOVO118, we have less RhoA activation. And so we wanted to test this in an animal model. And we started doing some contusion injuries in rats. We're using just a thoracic level 8 contusion injury. The red marks are where that injury is happening. And then really immediately after injury, we are installing a pump. And that pump is going to be delivering an intrathecal, so it's going to be delivering either NOVO118 or vehicle through an intrathecal catheter directly to that within the space of the spinal cord injury, so within 2 spinal levels. And it's going to deliver these for 38 days, which is the limitation of this system. And then we're going to look at weekly what's happening to those animals, and at the end we're going to look at some histology. And so our first bat at this was with J.K. Lee's group at the University of Miami, and you can see that there was faster and greater recovery based on the BBB subscore in animals that were given NOVO118 versus in red versus the animals that were given vehicle in black. Strikingly, most importantly, I think, is that these scores are represented over here on the right-hand side where, as we saw in the previous videos, really immediately after spinal cord injury, animals are dragging their legs behind them. After about 6 weeks in this particular paradigm, the rats are able to move their legs a little bit, but they waddle around more like a seal. And our NOVO118 treated animals, they actually have control of their paw placement. They're able to put them under their leg or put them under their bodies and have some support. Important to the spinal cord injury field in general, and while that was very exciting for us, we wanted to repeat it. And so we needed to do a replication study, mainly because we know that there's been many treatments for spinal cord injury over the years, and they haven't been repeatable. And so we wanted to be able to actually show that we can repeat this with another lab and with another group. And so we found John Gensel at the University of Kentucky, and he repeated the study, the same paradigm. We just took this out a little bit longer. And you can see again that NOVO118 treated animals had greater recovery based on their BBB scores, their BBB subscores. And this plateaued about 5 or 6 weeks. Another thing that I want to point out here is that you might be noticing that this effect doesn't look quite as good as the effect we saw on the previous slide, and that's true. But I think we want to stress that this is an important milestone for Novoron because we have been able to show with a different lot of drug, with a different group, a functional recovery. And as a result of this, we've been kind of banging our heads against the wall, trying to figure out why we're actually seeing this difference between our two different studies. I want to point out here that one of the problems has been variability in the functional recovery of our Novo-118 treated animals. And our current hypothesis is really that this is based on variation due to drug delivery. And so I don't think I've been clear previously, but our drug is a protein, so it's a biologic. And as an example, so all of the animals in our vehicle controls, those pumps stayed together. So there's a pump connection the pump gets connected to the intrathecal catheter. It stayed connected, and more than 90% of the vehicle was delivered to all of our animals. In contrast, what we saw with our Novo-118 treated animals was that connection came undone in many animals, and the reservoir volume basically showed that about 50% of the drug was actually getting delivered. And so even despite this variation that we're seeing, this average right here is including all those animals. And so that is still, we're still seeing statistical significance, but we do have this problem of drug delivery, right? We also wanted to look at some basic axon regeneration. This is just a serotonin density staining analysis. And so serotonin is a fiber that basically would come from the brain. And so after severe spinal cord injury, those serotonin fibers get disrupted, and there's going to be less of them. And so if we're actually seeing any sort of increase in axon regeneration, we would expect there to be a higher density of serotonin. And you can see here that if we're comparing our vehicle to Novo-treated animals, there is a trend potentially for more serotonin in these animals. And I just want to point out that over on this side, if we pull out just the Novo-118 treated animals, and we're looking from the epicenter of the injury to caudal, so going away from that injury, we see kind of like a bimodal distribution. And if I just separate out the animals that had intact delivery system and had delivered more than 65% of the drug, we see that we kind of have a group of responders and non-responders. And so while this is still exciting, I think it's also important to point out that this system is not going to be compatible for our IND-enabling studies. There's too much variation if we're going to start doing any kind of maximum tolerable dose range finding, therapeutic window type studies. This type of variation is just not going to be great. And so to that end, we have developed and validated a new delivery system. And so what I'm showing here is a rat-tethered drug delivery system, where we're basically putting an intrathecal catheter and we're connecting it to an external pump via a connection on the animal's skull. And so this does two things in terms of being able to keep the drug outside of the animal, because it's a protein, and we already know that risk factors for aggregation and precipitation of the drug are temperature and agitation. And so it's not inside the animal anymore, so we're not incubating at 37 degrees, and that animal's not moving around with the drug constantly, basically increasing the risk of the drug falling out of solution. And then two, we can evaluate patency of this catheter in an animal in the middle of an experiment. And so this video is just showing we can place contrast agent into that port, and we can look at how that's actually being delivered into the animal using a micro CT. And so at the end of this video, you can see that at the tip of that catheter there's a little cloud. And so this is how we're actively been able to test patency in these animals. And so we've validated this two ways, really. One, we've tested flow rates in our animals, and we found some things. And then we've also tested multiple vehicles. So we've been testing different formulations of the drug, and we've been able to do some tolerance studies with that. When it comes to a large animal like a pig, or even clinically with a human, we also need a drug delivery system. And so we've been having collaborative discussions with a large device manufacturer, and this is just here to basically show that, again, so this is an intrathecal catheter that would be connected to a subcutaneous port that we could then hook up to an external pump. similar to what I showed on the last slide, but this is in a pig or in a human. And importantly, we're just basically showing here that our drug is compatible with this system. So we don't have any unexpected aggregation. We don't have any unexpected sticking to the inside of this device. And we also don't have any unexpected changes in activity. We've been fortunate enough to put together a very good advisory council. In the interest, so this is our advisory council, and I just wanted to point out that we've been working together with some very great individuals. And I want to bring us back to the targeted drug delivery. So I just talked about comprehensive antagonism, and the other side of the coin is a disruption of a single ligand and subdomain interaction while keeping the biology of the receptor in place. And so this is really just a highlight that LRP1, the same receptor we've been talking about, is also implicated in binding to tau and is involved in tau spread, which is a hallmark of late-stage Alzheimer's disease. Importantly, we've been getting some traction along this because we have identified a single point mutation in a single domain that when that point mutation is made, that actually blocks tau interaction with LRP1. And so this validates that not only are LDLRs able to be targeted specifically by receptors, but we're actually able to go back and precision target a receptor ligand interaction within this system. And so we are in the middle of a fundraise. These are just some of our program timelines. What I want you to really take away is that while we have uncovered some problems with our drug delivery, importantly, we've been actively applying innovative solutions that will better inform our drug delivery paradigm when we actually get to a clinic, into the clinic. And so you can see here that for acute spinal cord injury, our program, we have a lot of work still, but we are using some innovative tools in order to actually get this work done, and we think to produce a better data set for actually being able to deliver it to a human when we get there. So this is just some key points to take away, and really who to contact if you want to contact us. So thank you for listening.
Thank you for the presentation, Tom. Very interesting approach here. I guess my first question is around the administration and the placement of these ports. How much training is that going to need from the perspective of, like, is this a novel surgical procedure when you start to move towards a clinic, and how rapidly would you be looking for the placement of that catheter?
Oh, two very good questions. And so the groups the device manufacturers that we've been working with, and then also some of the clinicians we've talked to, placing an intrathecal catheter is not something that's outside the wheelhouse. And then this port could really go anywhere. The main suggestion is that it would go kind of on the chest, and then that would be hooked up to some sort of outside pump. In terms of the timing for this, right now we want to target within like 14 days. We're really talking about a myelin inhibitory environment, and so the biology has to match. And so it's acute spinal cord injury. Probably the catheter would be placed around the same time as a decompression surgery.
Great, thank you so much.